So our case is case one, and our topic was gene therapy and CRISPR. Our group members were Parthivi, Chauhan,
Angie, Beato, Aishwarya, Billakurthi,
and Nathan Joseph.
So some people are opposed to gene therapy because it tampers with human genes, and critics are against gene therapy and they fear that gene therapy for disease may also eventually lead to gene therapy for more trivial and superficial matters like eye color or hair color.
So what actually is gene therapy? Gene therapy is basically a technique that is used to treat or prevent certain genetic disorders, and its initial goal is to do that while also avoiding drugs or surgery. Gene therapy is mainly used on disorders that have no known treatment. So there's multiple ways to edit genes. You could add a functioning gene into a patient's cell, you could replace a mutated gene with a functional one, or you could just go all out and inactivate the whole mutated gene. So the main type of gene editing that we focus on is an application called CRISPR, and CRISPR is an acronym and it stands for Clustered Regularly Interspaced Short Palindromic Repeats. So these repeats are basically sequences that are found in the DNA bacterium, and these repeats are usually copies of small pieces of viruses. These copies sort of act like mugshots in a way because they're used to identify viruses and other cell invaders.
A component of CRISPR that we will be further discussing is the enzyme Cas9, which is used to cut DNA. So going back to that mugshot analogy, bacteria can use these repeats to identify viruses, and then they can use Cas9 to destroy them. The CRISPR/Cas9 tool is very effective when editing genes, and many researchers have already used it for many different things, like fixing genetic diseases or combating viruses. CRISPR/Cas9 can also be used on human cells, and it could potentially disable harmful genes and cure some genetic diseases.
There are two main parts to CRISPR, and that is the Cas9 enzyme, which cuts DNA, and the other part is a type of RNA called guide RNA or gRNA. So gRNA has a specific sequence of bases on the end of it, and this sequence is called a target-specific sequence. It basically tells the Cas9 enzyme where to go, and it directs it to the specific locus on the DNA. So the Cas9 and gRNA combine to form a Cas9/gRNA complex, and this complex uses the target-specific sequence to find its destination. Here you can see that the complex has found its locus. This specific locus on the DNA actually has a name, and its name is the original target DNA. So the target-specific sequence on the gRNA binds to the original target DNA, and when they bind, Cas9 cuts the DNA and forms a double-stranded break. So this slide is just recapping the last few pictures, and one thing to keep in mind is that gRNA is very easy to make, and since it's easy to make, it's also very easy to program where we specifically want CRISPR to go and the specific places it needs to edit. So this slide is also just a recap, and after the DNA has been cut, now the DNA can repair naturally, but some things will affect it so that the gene will either be inactivated, deleted, or corrected.
In some cases, a process called non-homologous end joining results in the addition or deletion of a few base pairs, which disrupts the original DNA sequence and can cause gene inactivation. A larger fragment of DNA can also be removed by using two different guide RNAs to target separate sites on either side of the desired deletion. Cleavage occurs at each site, and the repair process joins the separate ends, thereby deleting the intervening sequence. Corrections to DNA can also be made by adding a DNA template along with a Cas9 guide RNA complex. The template is designed with sequences that exactly match the DNA adjacent to the target cut site. Through a process called homology-directed repair, the cell uses the template to repair the break, thereby replacing the faulty DNA sequence or even inserting a new gene.
So the three forms that the video talked about were disruption, deletion, and correction. For disruption, NHEJ occurs after Cas9 cuts the DNA, and this leads to a disruption in the DNA sequence and eventually the inactivation of a gene. Another form would be deletion, and this is used when you need to delete a larger fragment of DNA. The last form would be correction, where a template is added with the Cas9 enzyme to correct or insert a gene. The figures on the right help depict each of the forms, and the top one is disruption, the middle one is deletion, and the bottom one over here is correction.
Some other forms of gene therapy are somatic gene therapy and germline gene therapy. So somatic gene therapy is when new DNA is transferred into somatic cells, and this cures a disease in patients, but it doesn't prevent the passing down of the genes. So a patient who went through somatic gene therapy could potentially have offspring that have the genetic disease. Germline gene therapy, on the other hand, is when DNA is transferred into gametes, and this alters the gametes and prevents the offspring from having the defective gene.
So a little history about gene therapy is that in 1990, the first approved human gene therapy clinical trial was initiated. This clinical trial considered successful because it was greatly improved the health and well-being of few individuals who were treated during the trial. This was because along with the gene therapy, the patients also received their traditional drug therapy. However, the success of the therapy was tentative. This made it difficult to determine the true effectiveness of the gene therapy on its own, as distinct from the effects of the more traditional therapy. Genetically altering sperm, eggs, or embryo and initiating a pregnancy with them has not been shown to be safe or effective yet. Many believe it is not right to experiment with human life just yet. We see this genetic alteration as a stepping stone towards an unhealthier beauty and societal standard. Gene therapy will bring more economic discrimination to our society. It will add to the already rising social divisions in our community. It would also add to the class difference.
So now we're on to the legal concerns that come up when talking about CRISPR. Oncology Times did an interview with Joseph Rietta, who's studying CRISPR. The quote on the slide basically says that he and his team studied the first FDA-approved human trial involving CRISPR in the USA. The study was done in 2016, and since then, these CRISPR human trials were put on hold. This leads us into one of the more major legal and ethical concerns, which is which countries will be using CRISPR and which countries won't be using CRISPR on humans. This heavily depends on the beliefs and rules of certain religions, cultures, or countries in regards to gene editing and designer babies. An example of this is China and America. China has already been conducting human trials and shows no signs of stopping. America, on the other hand, put them on hold because they're more cautious with using humans. Because of this hesitance, the FDA put a clinical hold on gene editing human trials. The function of the FDA is to regulate gene therapy research, and anything they deem harmful can be suspended. The IRB and the IEC also have the ability to approve a clinical trial before it is carried out. Any equipment associated with carrying out gene editing, aka gene therapy products, have to be regulated by the CBER, and before a clinical trial is even conducted, an IND must be submitted. The gene editing products also need a submission and approval of a BLA.
So some of the pros and cons to gene therapy include that gene therapy could be considered a new ray of hope for many. In the USA, as of 2009, 3% of congenital disabilities were recorded in all births. Since there are no cures for those disabilities, gene therapy intends to correct these birth defects. Another pro is that gene therapy's effectiveness are timeless. And by replacing a defective gene with a functional gene, this is usually a one-time treatment that leaves the person symptom-free. Lastly, another pro to this could be that gene therapy can be used for other medical purposes too. Currently, the focus of gene therapy research is to provide solutions for people suffering from disabilities and diseases, but in the future, it can be open to fixing vision and hearing. There are also some cons to this. One of the cons could be that there's no guarantee that gene therapy will 100% work. Gene therapy is still in development, and most experiments are done on animals, with hopes that the same success will be reflected on humans. Another con could be that gene therapy is a costly treatment option. Because of the sophisticated equipment that is needed, as well as the high-level expertise, such high fees may not be affordable to everybody. This may create a socio-economical discrimination. Lastly, the last con we would talk about is that gene therapy may be short-lived, and some cells may become resistant to it.
So CRISPR is definitely not being developed to its fullest potential, and in the future there may be more medical and technological advances. Some examples of this are CRISPR-engineered cancer immunotherapies, and scientists are already looking into another gene editing enzyme, which is Cas13. Cas13 works with RNA to make temporary changes in the genome. An interesting advance that may be made is bringing back the woolly mammoth, which is an extinct species. CRISPR will help simplify and streamline the process of repairing the degraded woolly mammoth DNA that has already been extracted.
So our take on this case is that gene therapy is the tampering of human genes, and the human genome is critical to reproduction and evolution in general. Permanently altering the human genome has too many risks, and we believe it is unethical if it is used for superficial purposes. We are against using gene therapy for trivial reasons. So circling back to the beginning of this case, we think that the fear of designer babies is justified because humans always take things too far. This may be both good and bad, depending on how you look at it. Humans taking things too far could be good because we like to explore every possibility of a certain situation, but it may be bad because just because we can do something doesn't mean we should. We are against tampering with the genes of healthy embryos for trivial purposes.
In conclusion, using gene therapy for trivial uses is unethical and it should be outlawed as a form of practice. The main goal of gene therapy is to eradicate genetic disorders without the use of drugs or surgery. And using gene therapy for shallow purposes like eye color or hair color directly contradicts the whole objective of it. Last but not least, here are our references.